And people will probably be trickling in for the next few minutes, but we will go ahead and get started. So this is the FEC Seminar Speaker Series for Spring 2023. And we have Dr. Sulliván here with us, and Nayeli is going to introduce him. Thanks for joining.
Thank you so much. So I would like to introduce Dr. Mažeika Sulliván. He's currently a professor of forestry and environmental conservation at Clemson University and the director of the Baruch Institute for Coastal Ecology and Forest Science. At Dartmouth College, he earned a bachelor's degree in anthropology and Native American studies. And at the University of Vermont, he earned a master's degree in biology and a PhD in natural resources. After completing his PhD, he worked as a postdoctoral research fellow at the University of Idaho. And he was professor at The Ohio State University and director of the Schermeyer-Olentangy Wetland Research Park from 2008 to 2022. His role at Ohio State included serving as Chief Diversity Officer in the School of Environment and Natural Resources and developing best practices for diversity and inclusion. He's also actively involved in scientific societies such as the Society for Freshwater Science, where he serves on the board of directors and is chair of the Society's Justice, Equity, Diversity, and Inclusion Task Force. Dr. Sulliván has served on the U.S. EPA Science Advisory Board, and in 2020, he led a policy piece titled Distorting Science, Putting Water at Risk in Science magazine, discussing the negative impacts of the Navigable Waters Protection Rule, which aimed to restrict federal protection of vulnerable waters, including streams and wetlands. He also was recently awarded a NOAA grant to work with the Coeur d'Alene tribe in Idaho, and this project will integrate tribal knowledge and wetland science for 6 wetland restoration projects to enhance tribal resilience, increase wetlands capacity to mitigate droughts, and will also implement a monitoring program to evaluate how wetlands reduce drought effects. He currently leads the Streams, Rivers, and Estuaries, or STRIVE lab here at Clemson, which studies the ecology and conservation of stream, river, and wetland ecosystems. And he studies how natural environmental factors and human activities influence biodiversity and ecosystem function, and how to apply those findings to conservation and management. He's published numerous peer-reviewed articles, received over $7.5 million in research funding, and conducted research in various regions in the US and internationally. He's mentored 20 Master of Science students, 7 PhDs, and over 50 undergrads. And on a personal note, I've worked with Dr. Sulliván for a number of years, and he's been an incredible mentor. His energy and passion for science is contagious and inspiring, and he continuously encourages his students to strive for excellence in their research and careers. With that, I'd like to introduce Dr. Mažeika Sulliván.
Wow, thanks, Nayeli. That's great. And I will say, first of all, maybe I should have Nayeli give this talk because that was fantastic. And secondly, we asked Nayeli to do this on the spot just about a short while ago. So thanks so much, Nayeli, for that very gracious introduction. And it leads in nicely to the first. Well, first of all, I would just say it's a pleasure to be here. And so much of what we do in science is based on the collaborations and the colleagues and the friends that we make along the way. And before I even start speaking more and going into the talk of today, I just want to extend acknowledgements to so many wonderful collaborators and students and research associates and postdocs, many, of course, whom are not on this list. It could be multiple slides, but these folks are folks that have contributed most directly to some of the work that I'll be presenting today. But really, it has been wonderful. I arrived here in August, and Clemson, I just want to extend my gratitude to everybody for being so open and welcoming to the Clemson family. Of course, we're down here on the coast, and folks at Baruch have been amazing as well. And it's just been, but it's great to be here. And I'm pleased to have the opportunity to speak to you all today. I've obviously put in a lot of effort and given a lot of updates and talks around the state of where we are at the Institute and some of the directions we're taking, but this is a nice opportunity to talk a little bit more in detail about my own research program and what I do as a scientist. And so Nayeli really covered that quite well, so I can probably give you the Cliff Notes version here, but essentially I am a broadly trained aquatic scientist. I work to really ask the question that is central to the lab and to the research we do is about how variability in both natural gradients and human disturbance really affect biodiversity and community organization and ecosystem function. So thinking about these different levels of biological organization, how they layer on one another and how there's interactions among them. And I work in watershed and coastal systems, so streams, rivers, wetlands, and coasts. But a lot of what I think about and what I'll be discussing today in some depth is about how these systems are connected. So connected across the aquatic land-water interface and connected across various water bodies. And that idea of connectivity really frames a lot of the paradigm of my research. And as Nayeli mentioned, one of the key pieces, and over time, this has really grown in terms of the way we've been able to see impacts from this, but is how do we apply the findings from those questions to on-the-ground management and conservation, restoration, and then policy as well. So I work across scales, which has been it's been a great career. I really enjoyed working in very dynamic ecosystems in the US and in many international areas. A lot of my work focused on food webs, networks, and the ecosystem processes that they affect. And some of the tools that I use include stable isotope biogeochemistry, network theories and models, and a lot of thinking about fluvial geomorphology and how the changes in these aquatic systems really reflect or integrated into the processes that we study. So just to give you a bit of a smattering, again, to kind of work across that natural to disturbed gradient, as many of us do. Some of the areas and projects that I've had and the types of questions in terms of the alterations, the ecosystem, and even global alterations that we think about are done a lot of work thinking about urbanization. As Nayeli said, I was at Ohio State before coming here. Columbus is a large city, gave a lot of neat opportunities for long-term work and understanding the effects of urbanization. One of the pieces that I'll be talking to you today about is the effects of artificial lighting at night, which has been a really interesting stressor to study and to contribute to our understanding of how that affects aquatic ecosystems. Worked with issues around contamination, so nutrient enrichment. Nayeli and a lot of folks in the lab have been involved in a large EPA project looking at nutrient enrichment in watersheds, but also done work with heavy metals and others. Thinking about invasive species, climate change, issues around droughts and floods. A lot of work in wildfire in the West. I spent my postdoctoral years in Idaho and got engaged in a lot of questions around the interface between water and wildlife and wildfire, particularly in watersheds and stream riparian areas. And then again, thinking about alterations to hydrology and geomorphology and hydrology and how those changes such as dams and dam removal and shifts in channel fluvial geomorphic dynamics really affect ecological processes. Those are just kind of the gradient of some of the types of projects that I've been involved with. But before I do that, I think it's nice to know a little bit about where I've come from, and Nayeli gave a nice overview in terms of some of the highlights, but she made it really sound smooth and easy, and I really knew what I was doing all the time, and that's absolutely not the case. And I think this is helpful for folks to hear, particularly I think for students to hear that there's multiple ways to get to where you want to go. And I won't give you my whole life story, but I will say that as Nayeli mentioned, as an undergrad, I majored in anthropology and Native American studies, and I was very passionate about those areas. And really, when I graduated, I had no idea what or how to do things, and so I actually taught public school, high school for several years. And as a Spanish speaker, I was able to go in and speak and teach Spanish. And I also taught some biology and even some history. But again, I really didn't know what I was doing. I came from my own background was coming from a family who this was sort of a new paradigm for them. So I kind of stumbled along and did some, during the summer, I did some research tech experiences. And then I started a master's, but it was not knowing that you could go to school and get paid and get supported for a master's. So I was teaching and I was waiting tables and I was coaching track and soccer and doing all these things and getting my master's. And And then it dawned on me like, oh, this is how we do this. I got some folks that were, starting to mentor a little bit in terms of how you go through that grad school process. And so I went on and did my PhD in the more traditional route, but it was a good learning experience. And I think a lot of us come into science in different ways, and it's important to respect that and understand that lots of different pathways can get us there. So a couple other quick things I've mentioned. During my postdoc, during my master's, I really worked on restoration in agricultural landscapes, thinking about streams and restoration. And during my PhD, I really was trained heavily in fluvial geomorphology and the ecology, and that really was what my PhD was about. It was the intersection of those in streams and rivers. My postdoc really expanded that and was really about thinking about these aquatic terrestrial linkages, and particularly around food webs. And as my career grew, I had more opportunities to be involved in policy. And one of the sort of a watershed moment for me was being invited to the US EPA Science Advisory Board in about 10 years ago, maybe a little longer, to serve as an expert around water body connectivity and how that linked and was related to the Clean Water Act, in particular to waters of the US, which are those waters that are protected, federally protected under the Clean Water Act. And that really propelled a lot of work around thinking about how my own research paradigm in terms of the work that I've kind of shared that I generally do, but particularly around connectivity, was able to really move the needle in terms of the public policy arena. And so I've been heavily involved in that area since then, but really increasingly advocating for scientists to play a more active role in policy by representing the science. The other piece I'll mention just before I get into the heart of the talk is that based on my own, sort of multi-ethnic and background, and then also going back to my early training in anthropology and Native American studies, and then some post-grad work in those areas as well. I really have been able to develop some programs around biocultural connectivity and thinking about connectivity in terms of multiple ways of knowing as well. And so, as Nayeli mentioned, I work with tribal groups, and I'll be talking about that a little bit today as well. And then in a broader sense, that idea of diversifying science has been a big part of what I do, increasingly over the last decade, but certainly has been on the arc of my career very, very important. And, I certainly over half of the students that have come through my lab have been from underrepresented groups, and even more so when we think about women, probably 70% or so. And so, really thinking about how we can affect those changes and broadening our conversation and the people at the table has been something that's very important to me. And I think certainly is something that I've seen as a high priority in FEC and at Clemson, which is heartwarming. That's a little bit about kind of how I got to where I am. And there's been bumps and course changes along the way. So I think oftentimes when you hear people give these talks, we think, oh, they have it all figured out. But, I think we're all still doing that. And my path has been fun, but certainly not straight. But today, what I want to talk to you about is really about this paradigm of connectivity. And I'm going to focus on watersheds, although I do think it applies more broadly to ecology and other areas of environmental science. But I'm going to spend most of the time in the next, half an hour or 40 minutes talking about this idea of land-water connectivity and connectivity of water bodies, and how how the field is advancing around understanding how ecosystem boundaries are fuzzy, and how the exchanges of materials and energy and organisms really make these systems tick. But I also want to talk a little bit, as I said in the last slide, about extending that idea of connectivity, but thinking about connectivity of knowledge systems. And I'll give you a couple examples where I've been involved in working with linguists and thinking about how we can marry linguistic and ecological data, and then thinking about indigenous and or local and more Western approaches to understanding systems and what that can tell us. And just to mention a few projects I'm involved in there. And then the other connectivity dimension I will mention as I go through is just highlighting some of the conservation and management and policy outcomes that have been derived from some of this work I'll share with you. And that gets at the notion of translational ecology, which is something that many of us do, but certainly has increasingly received attention as a really powerful way of conducting science. And really it's thinking about moving, well, it's two things really. One is really thinking about moving our science beyond the journal page, right? So when we think about the end of a project, yes, we absolutely need to publish and get that up to the scientific community, but what are the outcomes we can do on the ground? You know, it could be workforce development, it could be policy development, it could be on the ground conservation, and working with teams of stakeholders and folks in other disciplines, and policymakers, right, or decision makers, and diverse groups at the table to do that. So I want to talk at the end of each of vignette. I'll share with you. I'll talk a little bit about how and in what ways we were successful, or we still need to be more successful in connecting the science with those, I think, very important outcomes. So let me go ahead and start with this idea of land-water connectivity and some of the work that I've done here. And sometimes in talks, and I won't today because I can't fit it all in, right? But I'll show some early artistic renditions of landscapes. And when I mean early, I'm talking about even 16th, 17th century, largely by European painters. But it really is interesting how the view of streams and rivers, and if you think about watersheds more broadly, but they're certainly looked at as ribbons dissecting the landscape and as very separate entities from the terrestrial landscape. And in the scientific world, it was really fluvial geomorphology, so the study of how water shapes the Earth's surface, but particularly relative to streams and rivers, where that understanding of the connections between the terrestrial and the aquatic areas really started to form. But when we think about on the ecological side, that didn't come till a little bit later. And even at that, the focus was primarily on the importance of terrestrial matter, terrestrial organic sources of matter, organisms kind of fueling the stream. And so if you look at this figure here, this depiction, you can see lots of material, for example, from adjacent riparian zones, such as leaves and woody debris. Even arthropods and other insects fall into the stream. And the paradigm for a long time, and in some ways it still is that sort of these terrestrial landscapes fuel streams and rivers. They're getting a lot of their energy from these terrestrial landscapes, which is absolutely true. But what we're finding out is much more variable than some of the initial notions were set forth. One of the reasons, and one of the key pieces that I've looked at a lot, and a group of us, is really understanding these reciprocal linkages, that the stream is also providing lots of reciprocal and rivers and wetlands, and I'm using streams here as a model, but think more broadly about this. But these systems are providing a lot of nutritional subsidies and organic matter, carbon packets, if you will, to terrestrial organisms and food webs and landscapes. And this can come in many ways, right? And come through, predator-prey relationships, such as birds and otters feeding on fish. It can come from overbank flows, right? When you have high flows, streams and rivers access their floodplains and deposit a lot of nutrient-rich soil and organic matter in floodplains. One of the areas that I've spent a lot of time thinking about is these aquatic emergent insects. So insects in their larval stages, right, are fully aquatic, and as they mature and as they metamorphose into adults, they get wings, most species, and they disperse into terrestrial environments. And they serve as very important vectors of energy and carbon and nutrients and potentially contaminants to a whole suite of consumers. And these can have both positive and negative effects. And I don't mean good and bad, but positive in terms of population effects or effects on food webs, right? You can have sort of positive and negative relationships. And so we can look at this, and it's one area that the field really developed on was very small streams, right? And even small wetlands to help understand this, but larger systems were less well understood. And so one of the questions that, we looked at for many years really, but I'll distill this into one slide, is in a fairly large river system, so fifth or sixth order, flowing into the Ohio River, which then of course flows into the Mississippi. We had a whole suite of sites that we looked at for really many, many years. For different questions. But one of the pieces we wanted to know was, well, in these larger systems, what is the contribution, not just of the prey itself, right? So fish or insects, right? But really, what is the contribution of primary producers? So the aquatic primary producers to terrestrial food webs, to terrestrial consumers. And so phytoplankton, for example, benthic algae and macrophytes. Now, we all know that a raccoon's not diving into the water and eating phytoplankton, right? But the organisms that are feeding on phytoplankton that may be depredated by something else, such as crayfish, are then eaten by raccoons. And so these trophic linkages move that aquatically derived energy to these terrestrial consumers. And so we use stable isotopes to Bayesian mixing models and a whole suite of tools to help answer this question. But in the end, we found that, at least in this system, these shoreline spiders that build webs really near the water and they intercept these insects that emerge out, aquatic insects, we're relying on 50% of their nutritional subsidies from this suite of aquatic primary producers. Raccoons, 48%. These are averages. You can see, for example, spiders, the variability was 40 to 69%, but these are just the means. Raccoons, 48%, damselflies, 44%, swallows, 41%. We looked at beetles and others as well that were less, but the big takeaway here, and I think the real contribution that got people thinking a little bit more about these systems as not just recipients of energy, but actually donors of energy as well, right? Was really a little eye-opening in terms of the percentages of their nutritional subsidies, sort of their energy, if you will, that are coming from these primary producers, aquatic primary producers. Even less is known in coastal ecosystems, and certainly in estuarine environments or nearshore environments, we know that these mosaics have multiple dimensions of connectivity, right? So marine to freshwater, freshwater to marine, land to water. One of the ones that really hasn't been looked at much is the estuarine or water to land. Yet we know from freshwater paradigms that could be very important, just as I shared with you in the last slide. And so focusing on those emergent insects again as one way to help measure this, we worked for several years in many systems. The one I'm going to share with you today is the Rookery Bay Estuarine Reserve in southwestern Florida. But the question we were asking was, well, in this system that ranges from freshwater, right, to saltwater, are there actually, a quantifiably meaningful emergence of insects that are leaving these systems to disperse in terrestrial environments and provide this energy? Because a lot of the literature and the data are suggesting that there are not a lot of marine insects, if any, in these areas, and so the thought is that these are going to be pretty depauperate in terms of those populations, right? And so we worked in this area, and I've worked in others as well, as you can see here. But the one I want to talk about today is this, 10,000s area. It's in Southwest Florida. It's mangrove dominated, but again, freshwater. We worked in freshwater to sort of mid salinity to brackish to polyhaline, highly saline sites. And the first question we asked was, well, what can we just survey these insects that are leaving the water as adults and look at that variability from the freshwater to the polyhaline or the high salinity environments. And so in a nutshell, we found 20 aquatic insect families from 6 orders. And just as a point of comparison, we found 27 in that Ohio River that I was sharing with you. And they came, from most of them were these non-biting midges, which are fairly tolerant. So that didn't surprise us. And the emergence rates, and we collect this by putting these little floating tents over the water where the insects emerge out into, so we can get an idea of the rate of emergence over time. You can see range from 0 in some sites to 68.7 per square meter per day at other sites. But the mean kind of fell out within that mean that we were seeing in the river. So lower, but still, I think, of quantitative importance. And we can see here, and these are, ordination plots that depict the emergent insect community composition at the top by salinity level and at the bottom by season. And I won't go into too much detail here, other than to say that we saw that the community composition of those insects shifted across the salinity, which we would expect. And they shifted based on the seasonal pattern of wet, dry season. So both of those were something that made sense ecologically based on the life history of these organisms, but put a quantitative estimate around these observations. And so we can look here briefly again at some of the differences, the variability that we saw in some of the metrics for these insects. And so panel A is emergent insect rate. So right, the rate at which these insects are leaving the water and emerging. B is the number of families, C is a measure of diversity, and D is a body and a mean body size. So, and that's really a trait that we think about as, many traits, but one is important to help us understand the extent to which these insects can be important nutritional subsidies for terrestrial food webs. The statistical differences are those which do not have any shared letters in the code above them. So A is different than B. So in this case, in the winter, we saw emergence rates were different between low and mid and high, but not between low and mid, right? So this is where you see the difference. So again, lots of different patterns here that we could spend a lot of time deciphering, but for today, suffice it to say that there were patterns that were expected based on the seasonality and the life histories of the insects, and some that were more surprising. Certainly, we can see here that these data support that insect mediated subsidies to terrestrial zones in these estuarine, even at the high salinity sites, so polyhaline sites, can be important during certain times of the year. But the linkages with the terrestrial consumers is we really need to make that key link, right? And so what you're seeing here is essentially looking at those spiders again as a model terrestrial consumer. These weren't tremendously strong relationships, but enough to suggest that yes, these spiders were feeding on these insects both in the winter and in the summer, and that was a key vector. What you're seeing at the bottom is also a positive relationship, but this is by salinity. But as I said before, and a key piece of this is that aquatic insects don't necessarily equal aquatic energy. And let me just repeat what I mean by that, is that aquatic insects, for those of you who work with them or know them, recognize, right, that different species, even at a higher, different species, different even at the generic level or the family level, right? We know that there's gonna be a lot of variability in what these organisms eat. And so some are gonna be feeding completely on detritus, so terrestrial organic matter that falls into the water that they then break down and feed on. Others are going to be feeding more on aquatic algae, for example. Some are going to be feeding on a mix, right? And so what we're talking about for aquatic energy is those aquatic primary producers, not the terrestrial energy that's sort of recycled through the aquatic system. And so one doesn't always equate with the other. And so this is where we use these isotopic approaches again. And this is just an example of a plot from across our freshwater sites and these estuarine, or this estuarine gradient, where our nitrogen values are on the y-axis, and the carbon values are on the x-axis. And essentially what this can show us is, here's our consumer, our different types of shoreline spiders, and here's our major aquatic insects that are leaving the water. But what we can see for this is that in this case, tetragnathed spiders are relying on a combination of sources, and including a combination of phytoplankton, epiphyton, and terrestrial vegetation. But that doesn't quantify it for us. It just gives us a visual idea of where we can parse out the nutritional subsidies. So again, we use Bayesian mixing models, and this is small. I recognize this, the panel on the right, bottom right-hand corner. So I don't expect you to dig into that. But briefly, I would say that what I would like you to kind of take a look at is that this is all phytoplankton and epiphyton, right? So these are all aquatic primary producers. And you can see from 0 to 1, the proportion of the reliance of all these different types of spiders really is very high on these, highly reliant on these aquatic primary producer pathways. And so you can see that here, especially in the moderate level salinity, you're getting almost, over 90% reliance on these aquatic primary producers. And so this was really interesting, right, for us to understand that even in these saline environments or partly saline environments, that these aquatic consumers are still, or excuse me, terrestrial consumers are still reliant on these aquatic primary producer pathways. So sort of aquatically derived carbon, if you will. So that is just a little bit of some of the basic work in the aquatic sort of terrestrial linkages and helping understand the type of work that we've been doing to help quantify to the extent that it's not just organic matter fueling aquatic systems, right? But it's also aquatic systems fueling these terrestrial systems. But as I said early on in the talk, we also look at a lot of questions around environmental change, human-induced and natural. And I talked about more of a natural one in certainly, you know, ranges of salinity. But we are changing our Earth in so many ways. And so these linkages and these ideas of connectivity might be expected to be altered as well. And I think if we were extraterrestrials, right, aliens flying over the Earth, the first thing we would probably notice is that it's glowing, and this was not always the case, right? So certainly in an evolutionary time frame, it has been just a blip in time that the Earth has the lighting patterns on the Earth have been altered. Yet all species, including humans and all ecosystems, have evolved along that with a predictable light dark night day pattern, predictable seasonally, right, and predictably in terms of predictability in terms of the life history strategies of organisms, reproductive strategies, behavioral mechanisms, and so forth. But as you can see from this map of the U.S. Or U.S. and Canada, well, actually all of North America, you know, it's the lighting is extreme, and we're certainly no exception here in South Carolina. And so over the last decade or so, we've been looking at this question relative to these aquatic terrestrial linkages, as well as other questions in community level questions and ecosystem function questions. But how does this lighting really interrupt these really critical connections? And we've done that through a suite of surveys. You know, when we go out and we actually look at sites, streams and wetlands that are exposed to lighting in urban environments, but we also do that with some manipulations. So here's a wetland complex where we added light, right, to have a manipulative component where we could really test and constrain some of the other variables that we wanted to constrain. And so I'll share a few of the responses. And so in streams, we found in this particular study where we looked at a suite of urban streams around the Columbus area, that these shoreline spiders, and again, I'm trying to keep with the theme here so we don't have to create, well, so you can kind of follow some of these consumers that I've been talking about. But these shoreline spiders, their density decreased by 44% in these highly lit areas. And to give you a sense of what this lux means, the low lux in this particular study was 0.1 to 2. It's really hard to find 0, right, in an urban environment just because of sky, you know, just the sky glow in general. But to give you a point of comparison, a moon, like a clear, if you go out on a clear night and then there's just a moon out, a full moon, it would be around 1 lux, maybe a little bit less. And so, or sorry, that's just moonlight in general. A full moon, you know, we can think around, you know, 0.25. So, but either way, 0.25 to 1 lux, you're getting the idea. It's, you know, this is pretty low. And then these are what we would find under street lamps or, you know, near them, at least in riparian areas. But we also saw, you know, that these were linked to these emergent insects. So these emergent insects decreased by 16% under lighting. And the diversity shifted as well. Interestingly, when we look at the terrestrial insects, so those in the riparian zone that are terrestrial, you know, feeders like beetles and sow bugs and a whole suite of terrestrial inverts, the body size increased by 309%. So these huge differences in body size. And that's not saying that species A got bigger. It's saying that new species came in that were larger and largely predatory species. So real big differences in these communities under the lighting. In the wetland, we saw that emergent insect, both emergent insect, aquatic emergent insect density, as well as changed by both the lighting intensity, right? So we saw a difference between our control and are moderate and high lighting. Again, you can look at the levels. 10 to 20 is high, but not more what you'd find with under a street light, for example. But we didn't really see, we saw there's a difference under the light emitting diodes and the halogen type lamps. So two different types of lighting, and the LED are really relevant because that's across the US and many parts of the world. Europe, for example, a lot of the halogen, the old lights are being switched out with these more energy efficient LED. But the question is still out on whether these are actually ecologically beneficial. We didn't really see a difference there. But we did see in this ordination plot shows this, that the community composition of these emergent aquatic insect communities shifted under the different lighting spectra. So the different, these, right, so the lighting spectra are which lights are like, you've the blue light emitting diode, so the LEDs are more of the blue light emitting diodes, right, with the HPS, the spectral composition of the light itself is going to be a little different. So we saw differences there based on some species turnover. Going back to the streams, if we think about more of a functional response, food chain length is a measure of from the basal source to the top consumer. And it's a really important food web, measure of food web architecture, but also tells us a lot about ecosystem function. And so we saw that invertebrate food chain length was lower at these moderate and high Allen sites, so Allen being short for artificial lighting at night, than it was under the control. With the implication that as we think about not just shifts in community function, and I think in this case there's multiple reasons that we could think that food chain length would be altering some of these functions, or as a reflection, I should say, of some of these functions. And so thinking about, I mentioned before, as we talk through some of these, what are some of the conservation policy implications? And so the work on artificial lighting at night certainly is something that I'll be engaging in here in South Carolina, in more of some coastal environments as a major key question in coastal areas where we have tremendous development. But we need to balance human and ecological needs. And so when we make recommendations, it can't just be leave it dark in areas where humans need to be. But to make recommendations, we need to be able to make recommendations on lighting type and the duration and the direction. Came about 5 years ago when the Department of Transportation, the Ohio Department of Transportation, funded a fairly large project. It was $600,000 or $700,000 for us to answer some of these questions for them to build into their actual engineering. And so we're just finishing that project. And this is sort of a brief infographic that we're giving to the state, and that they are really willing to make some changes based on these recommendations. And so based on the lighting level, where we think it's going to be most impactful, one of the pieces that we found for fish, and I didn't share this today, but was that strobing lights from some experiments were really interfered with feeding, predatory feeding. And so when you think of coches, like the cars driving over bridges, right? They often the rails on the bridges are slatted, right? And so the light has this strobing effect. And for example, that is something that maybe a recommendation, let's try to avoid those. So really bringing this into on the ground on conservation, but policy around best management practices is an example of where I think this can be very impactful. So switching gears a little bit, another sort of ecological connectivity around land and water, but where we're seeing some really important ecological questions, but also conservation questions, is around aerial insectivorous birds. And so again, worked with this guild for many years. Aerial insectivorous birds are so important for ecosystem functioning in terms of insect control, pest control. They are a multi-million, if not billion-dollar asset to the agricultural sector, and there's so many reasons, that they are just critical to not just ecosystems but human well-being. But we're seeing these marked declines in aerial insectivore populations, so barn swallows, swifts, nighthawks, martins, right? And so, but we're not sure why. But this is happening across the guild and very concerning. And so we have been looking at multiple questions around aerial insectivorous birds in riparian areas, and that's the key link with the aquatic systems, because they tend to be highly diverse in these riparian areas, largely because of critical habitat, but also because those aquatic insect sources that I've talked a lot about are packed full of really important fatty acids, really important lipids. And these lipids, and there's been really some neat work shown in other research groups to show that those aquatic insects are energetically much more valuable in terms of energy per mass, right, than terrestrial insects. And so these riparian areas, whether it's around wetlands or streams and rivers or coastal riparian, or even coastal riparians, are very, very important for the conservation of these birds. So the question we looked at was, well, we're also seeing urbanization as a major footprint, particularly in areas where we're seeing declines. And with urbanization, we start to see shifts in water quality, changes in the flying insects, right, the emergent insects, but also the terrestrial insects, and how does that play out? So we use tree swallows as a model aerial insectivore, and had multiple sites sort of for city birds and country birds, right? So we had sites within the city and sites within in the rural areas. And I'll just share some really interesting highlights that may challenge some of your expectations of what you might think here. So we actually found that urban nesting tree swallows, and this was a multi-year, 5-6 year study, initiated laying almost 8 days earlier and fledged 35% more young per nest than those at protected sites. Those are really meaningful. So 8 days earlier is a big deal when you're talking about birds that have an 11 or 12 day incubation period, right, and a few weeks in the nestling stage. We saw greater reproductive success in urban settings where aquatic insects tended to be larger in body size. So the species tended to be larger bodied species. Local climate conditions were more amenable to egg and nestling survival, and the breeding season was extended, particularly in colder climates where it buffered these really, the urban environments buffered some of the early season cold snaps that really can be detrimental for aerial insectivores like swallows. And then multiple characteristics of urban sites appear to drive some of these patterns. So mean and extreme air temperatures and measures of water quality, for example. But that's not the end of the story. The other part of this to think about is we also saw that and again, using isotopes, we saw that trophic position of adult swallows was higher, almost 10%, so a little more than 8% higher at urban than naturally protected sites. So a shift in trophic position. Now that might not seem like a lot, but it has ecological meaning. And where it really has meaning and where this gets complex is that there are trade-offs. Right. So even though that these urban environments appeared to advantage some measures of reproductive success, we also saw that there was a 482% greater adult swallow contaminant burden in blood, adult swallow blood. This was mercury at urban sites that protected sites. Now, the levels of mercury were still pretty low in these systems. So that even though it was, really a lot higher, right, magnitudes higher, it still, wasn't at such a level that it would, it was still a relatively low level. But nonetheless, that pattern, right, suggests that there's these really important trade-offs. We also looked at a global scale, a national scale analysis, and then we extended this out to think about, okay, well, what if we use the breeding bird atlas and think about water quality across the US, and can we match these up to help understand potential declines in these really key aerial insectivore species with overall patterns of water quality. And so what you're looking at here is they're called chloropleth maps, right? And I'm sure you've seen these in many different contexts, but essentially the darker, I'll go back here, 1 second. The darker colors here, the blues and purples are where you have higher, so they would be the higher relative abundance of emergent insects, right? So those that are potentially food sources for these aquatic, aerial insectivorous birds, and the red orange would be the lower abundances, and the top panel is streams and the lower is lakes. And so that's you can see that variability, right? So you can see here in South Carolina that we have among the streams are producing relative to others in the country pretty high rates of emergent, the relative abundance of emergent insects is pretty high in the insect community. But so we linked that with water quality, and we found, and then we linked, right? And so we found that the real drivers were ammonium, road density, percent sands and fines, so siltation in rivers and streams and lakes. Those really linked to the relative abundance of emergent aquatic insects. And what I mean by that is relative abundance, that there are a set of aquatic emergent insects that don't emerge. They don't ever leave the water, or they never make it to that stage because the water quality has been diminished. And so that is a measure that we were able to use for this study to give us an idea of potential food sources for the emergent the birds. And if we look at those aerial insectivorous birds, right, and these are the populations that we modeled based on the breeding bird atlas, and I can't go into this in too much detail today because we just are limited by time, but we look at these riparian, obligate, and facultative, and upland, and then non-aerial insectivorous birds. And the short story is here, we absolutely saw some important patterns with some key aerial insectivores like alder, Acadian flycatchers and olive-sided flycatchers. But we also saw patterns, confusing patterns with others. So it's a bit of a messy story, but certainly there are trends that suggest that at a national scale, we are seeing these shifts in water quality linked to these shifts in certainly in emergent aquatic insects, and then among some aerial insectivorous birds are directly linked to that. So if you think again of the conservation and policy, we can kind of go back to the idea of that I was talking about with the cities, is that there's trade-offs, right? So, some of the recommendations we've made to urban planners and others is sort of maximizing benefits and minimizing risks. So in cities, clearly, these can be environments where some of these species can thrive, but we need to minimize the risk of contaminants, for example. One of the pieces that we've been working on, on a larger scale is really with agencies and even funders, but management agencies, to kind of break down some of these aquatic terrestrial boundaries, because in order to conserve some of these species, we have to conserve those connectivity points. So I want to switch gears a little bit as we kind of round out, getting later in the talk here, to talk briefly about, I've talked about land-water connectivity, but I want to discuss a little bit the connectivity of waters themselves. So upstream to downstream, and even across landscapes. And what you're seeing here is a pictorial, just a graphic of multiple types of water bodies in a watershed, and how ultimately they're all connected in some way. This shows hydrological flow paths, but we can think about being them connected in chemically and biologically to downslope and downstream waters. And I'll share with you in a few minutes, this is really important to in the policy arena. And a quintessential example of this, of course, is when we think about nutrient enrichment at a large scale across the Mississippi Basin, as you're seeing here, and the dead zone in the Gulf of Mexico, which is, I think the last I read was the size of New Jersey, right? I mean, it's a large area, and clearly the nutrients, and we know this, certainly are coming from, urban and agricultural practices across the basin, not just down in the lower Mississippi Basin, right? And so that's just, a great example, a sad but great example of water body connectivity and how that plays out. One of the benefits I had at the last institution where I was director, it was a Ramsar wetland site, and there was a long-term data set. And certainly this was something that I benefited from. I did not set this up. This was in practice many years before I was on the scene there, but to be able to look at these long-term patterns is powerful. And we were, to look at, for example, this is a constructed wetland to see how these restored wetlands actually function. And here's a river, and there's flow throughs, a couple inputs, an input and a couple outflows through these wetlands. And so we just, for purposes today, I just wanted to show you an example from a few years where we looked at nitrate. Just put this together so as an example of how these systems can function. You can look at nitrate, and the inflow is in the blue or purple there, and the outflow is in the other colors. And so the inflow is where the water is coming in from the river with nutrients, right? And then the outflows are where it's leaving, also has nutrients, but much lower levels. And if we just do a quick analysis of this, we can see that the outflow sites are from, around a little more than 2 times the levels for nitrate than they were coming in. So these can be very effective, even these restored or ecologically engineered wetlands. And we see very similar patterns for phosphate and turbidity as well. And we have a new paper that just came out this year, just hot off the press, that we are with some colleagues coming up with some phosphorus retention models for these inflow outflow wetlands to be able to model the amount of phosphorus that can actually be stored in these wetlands. So that's one example of these sort of water-water connectivity. Another is the way organisms move nutrients and other material between disparate wetlands. And so I want to show you this example because it was kind of a fun project to be involved in, and I think hits on this connectivity in multiple ways. But two ways here. One is we looked at waterfowl across water birds across multiple wetlands and distributed in southern Africa, South Africa being one of the countries, but Botswana, Zimbabwe, Zambia, Mozambique, others. And what we essentially were looking at is the movement of these largely for waterfowl and water bird conservation. Could we actually use the isotope ratios of these carbon, nitrogen, oxygen, hydrogen to track and to understand where the molting origins were, because these are such critical source areas for these birds. But it also gets at this idea of how important these wetlands are in terms of these organisms are in moving biological material and connecting these wetlands. So I won't go into lots of details here other than to say that we did see we did provide evidence here that at least a subset of these isotopes were very valuable in helping understand what locations. And it was just a neat project. And I think that idea of translational ecology, really thinking about, we worked with multiple teams, and I was not the lead on this. I was brought into this, which was great. But to be able to work with teams of folks, even to think about disease like avian influenza and link it to human communities and risk was very interesting. And the last little piece I'll share in terms of water body connectivity is this idea of upstream to downstream. And one of the key pieces here is understanding that our nation is there's a really interesting video, a documentary called Dam Nation. And I think the pun was meant to be there. We're sort of damming our rivers in both the literal and the figurative way. And again, South Carolina, not an exception here. And so we've done a lot of work understanding the effects of dam removal as a tool for restoration, but what actually happens when a dam comes down. A lot of these have been here for decades, if not generations, right? And so these systems have fundamentally shifted. And so we had this neat opportunity two urban dams were removed to look at the effects of dam removal at multiple levels of ecosystem contamination, biological responses, terrestrial land-water connectivity, water-water connectivity, just a lot of different ways. And so I'll just share this neat story in a very brief story, is that we did see in these systems, certainly in the initial years, a general coarsening of the sediment and a redevelopment of riffles, which was neat to see, as we might expect, improvements in chemical water quality, and then changes in biodiversity. But where I want to share a little bit is that idea of food chain length again, and this focused on fish. And if you look, and this is just a few years into it, and I think the neat story will be in soon. So I'll get back to you because we have 10 years of data now, and we're working on a 10-year sort of analysis of this. But this was just a few years. Initially, you see that bass and carp are your top consumers before the dams were removed. And then the year after, the carp stayed. And we started to get more sunfish as the top consumer. And these are your trophic positions here, sort of food chain length rather. And then the year 2, we started to see darters come in and other small insectivorous fish, and carp fell out, right? And then year 3, we started to see some bass come in, but feeding at a lower trophic level. And so really interesting in terms of how these communities reshuffled after based on these environmental changes. But one of the interesting story here is that we also can link food chain length, and I said it was really important of ecosystem function. Well, one of the reasons is because it's linked to contamination, right? And so the higher the food chain length, likely the higher biomagnification. And we looked at dieldrin and aldrin and other pesticides, as well as PCBs and so forth. But the short of the story is that the contaminants didn't really change in our abiotic compartments. So the water, the sediment, even though they were redistributed, the contaminant concentrations didn't really change that much. But the overall ecosystem contamination shifted because it went down, right? Because our top level consumers came down. And so we had just a lower capacity in the biological compartment to biomagnify those contaminants. So that was kind of an interesting story there. And that's been this work has been really interesting. We've been fortunate to get on NPR and others to talk about dam removal and to think about some of the pieces we've learned in terms of informing restoration efforts. So I know I have a few minutes left, and I have a few things I would just highlight. I think on the ground pieces have been really important. Part of this work has informed some of the restoration, on the ground restoration work we did. And so, we have propagated about 6,000, individuals and multiple species through translocation projects that are linked to some of the previous funding that I had gotten with in Ohio to actually do some of the on the ground conservation. As I mentioned, a lot of this work directly links to policy around connectivity, and there's been some really important avenues, and I would encourage students and others to think about how you can get involved as a potential scientist, a budding scientist, or in your career as a scientist, if that's where you go, to be impactful there. And then the last little area I would talk about before I wrap up is that these ideas of biocultural approaches to conservation, I could have a whole talk on that, but I just wanted to put a placeholder in here for future conversations that I think in my work and others that conservation really must draw on and find the interface among these multiple knowledge systems. And where I've been very fortunate is to work with tribal folks, developing relationships for many years, working with the Coeur d'Alene tribe in Idaho, or what they would certainly call current day Idaho, on issues relative to water rights and the amount of water that is necessary to support traditional uses of hunting and fishing and gathering. But also to, more recently think about, we had, Nayeli mentioned we have a new grant partnering with this tribe, and there's a suite of these across different tribes to think about how we deal with droughts, particularly increasing what we're seeing increasing droughts in intensity and frequency. And so this is a really neat project to blend Indigenous and Western knowledge. And to think about that, we have a paper we're working on, and this is, I think, is how this translates to on the ground work. This is in review right now to think about how do we enhance protection on tribal lands and think about connectivity and what that means from a tribal standpoint, not just from a Western standpoint, and how that should be linked into policy. So I'm going to go ahead and wrap up. I don't have time to talk much about these pieces. I have a couple other projects thinking about biocultural approaches to conservation in Lithuania and Eastern Europe and the Amazon. But overall, the arc of what I do, and certainly where I have worked for years now, is to think about these multiple dimensions of connectivity, both in terms of a pure scientific standpoint, but also in terms of broadening our ideas of connectivity and the ways we think about doing science, the partners we work with, broadening our communities of practice and our audiences to really be effective in terms of that idea of translational ecology, or just really that the science that we do reaches and has the effects that we hope it does. So thanks so much. Muchísimas gracias. It was great to talk to you all. And I know we're at the end of this time, so I'll open up to any questions, but I can also stay on for a few minutes after if folks just want to chat. And I put my contact information here, and I'm always happy to engage and meet more folks. Again, I've only been here a short time, so looking forward to continuing to get to know folks. So thank you very much.
We have one question coming in the chat here from Dan Knapp. Do you think that amphibians have a potential to contribute to land-water connectivity? And is it possible to incorporate them into your estimates of energy export out of freshwater systems?
So thanks for the question. And the easy answer to that is absolutely. I mean, there are so many different vectors. And what I shared to you today was a little snapshot, just focusing on the insects mostly, but we've also not only looked at different vectors, but different responses. We looked at, looking at lizards, we look at birds, we look at mammals, but certainly in terms of the aquatic vectors, I was on a PhD committee recently looking at salamanders and trying to make some of these estimates. So absolutely the same tools can be applied to a variety of different organisms and to help, quantify those estimates that I think you're interested in. So absolutely, and always happy to, find the time offline to talk to you about that.
Feel free to unmute yourself and ask a question or type it into chat. Dan says thank you.
Hi. Hi.
Hi, we hear you.
Yeah. So I had one real quick question. So thank you for the wonderful presentation. So you had a wonderful combination of everything like ode and pictures. It was very nice slides over there. Thank you so much. And I was just curious about like, uh, you talked about the Elan artificial lightning at night. So how is it? I'm just curious about what could be the reason for the body size increase of the terrestrial insects and like while decrease in the aquatic ones, and how it is related to the decrease in the numbers of the aerial insectivores birds. That's my question.
So let me just make sure I'm understanding the question. So were you asking what is the mechanism with lighting decreasing the energy subsidies to terrestrial consumers? Is that what you were asking?
I'm asking about how is the lightning related to the body size increase of the terrestrial insects, and like is it related to how decrease in the aerial insectivores birds? How is it related?
Yeah, so in terms of terrestrial insects, if that's what you're asking, the lighting advantage is what we think. So sometimes there's these mechanisms we think of insertion or deletion mechanisms in food webs or communities, and insertion being where new species are added in, and deletions being when species are removed. And so lighting, we think, for at least the terrestrial insects, is advantaging some of the larger bodied communities that come into these areas because they're more lit, they have more opportunity, they're more effective predators there. So large bodied beetles, for example, come in. In terms of the aerial insectivores, we haven't made the link between lighting and aerial insectivorous birds, although we have been working on that question with bats. And so one of the things we see with bats, of course, is that lighting advantages some species, but not others. So those that depending on their foraging mode, certain lights attract insects, and then the bats, of course, are attracted to that. But it's too early to say. I can't tell you beyond. We're still doing sort of the species level analysis, but right now we can see that there's a shift in sort of the community composition of the bats that we're getting in these higher lit areas. I don't know if that quite answered your questions, but maybe if not, we can send me a note, or we can talk and follow up so I can be more specific in an answer to help you out there.
Okay, thank you so much.
I see your question, Casey. What are the thoughts for cities to incorporate red lighting like along coastal areas, or is red lighting more for less populated areas? Yeah, so I think I'm part of a group. It's an international working group on sort of lighting right now that's trying to bring in engineers and scientists and practitioners to answer these types of questions to scale up. One of the things to answer your sort of question, so I'll say two things. First of all, one part of the work we had to do for the Ohio Department of Transportation was to do a sort of a lit review, and it was a massive amount of literature, largely in the last 10 years that are coming out on lighting, which is great. But one of the pieces that is tricky is, I would argue, at least from my research, and I think talking to colleagues and just spending a lot of time thinking about this, that as a first pass, when we're talking about protecting ecosystems and multiple species, or an umbrella of multiple species, versus trying to protect an individual species, right? That we want to focus more on the lighting intensity, and then as a first pass, because that's really what seems to have the greatest impact across species and the ecosystems, if that makes sense. The reason I say that partly is because the spectral composition is so species specific, or even taxon specific, depending on how organisms perceive light, depending on their physiology, depending on their behavior. And so when you start talking about spectral composition, red lighting, for example, may be great for some species, but it could be very detrimental for other species. And so it really depends on what you're trying to preserve, right? What the conservation focus is. And then and that conversation has to be part of, what's the key priority. So I know that didn't answer your question about red lighting particularly, but I think it's just hard to say that, because it would have to, what's your conservation focus? I think really needs to be part of that question. But great question, and something certainly that we're all struggling with. Yeah, absolutely.
Yes, Ibera, do you have a question? Is that what your hand's up?
Yeah, thank you, the presenter. My question is, how does contaminants affect terrestrial food web?
Yeah, so I think in a few different ways. One of the questions we've looked at a lot, and we actually had a focus article about this, was about the mechanisms moving aquatic contaminants to terrestrial food webs. And so, and that can happen in many ways. And aquatic systems, as you are very much likely well aware, can be repositories for contaminants in multiple ways. So certainly they can collect contaminants in the sediment that would be coming from upstream. So dams are a good example of where we see oftentimes high concentrations of contaminants in the sediment from legacy, right, from activities upstream that accumulate there over time. But also there's atmospherically deposited contaminants like mercury that we even find in Arctic, what we would call quote unquote pristine types lakes, right? So or systems. And so the contaminants can move out of those in multiple ways, and they largely move out of them through biological pathways, so through food web pathways, right? So organisms pick up the contaminants in the sediment and they move those through the food web. So it depends. That idea of food chain length and the complexity of the food web in the aquatic ecosystem really mediates the biomagnification and the concentration or the actual amount of contaminant that then moves into the terrestrial system. And so those vectors like insects and fish and other organisms that are fed upon by birds and lizards and bears and all sorts of terrestrial consumers, including humans, right, in terms of fish, are the mechanisms that move it. And so it really is a multifaceted question where we have to think about the sources of the contaminants, where the contaminants are distributed in the aquatic environment, the food web complexity around how much is going to be biomagnified, and then the capacity for those food webs to have vectors that actually disperse and how far they're going to disperse into the terrestrial landscapes. So it's a great question, but that is sort of where we've been spending a lot of time thinking about putting numbers on that to get to quantify that in a better way.
Okay, thank you.
Okay, there's one more in the chat. I am going to stop the recording. Any students who still have questions are welcome to stay on. I think Dr. Sullivan said he had a few more minutes that he could talk with us maybe a little more formally. But if you need to get to your next class or meeting, thank you for joining us.
Thanks, everybody. And yeah, so Pravina, I can answer your question.